Role of periodic stress in the emergence of life. Today, many probiotic models have been proposed, which are considered as precursors of primary living cells. As an example, this is the model suggested by several Japanese scientists. This is giant vesicles with containing incorporated DNA. And then next step is amplification of DNA during the oscillations of temperature, and then division of giant vesicles along with amplified DNA. This is the same process in follicles, and after that, the following evolution of this model is over. So this probiotic model, as well as other kinds of probiotic models, do not display the following key properties that demonstrate activity of living systems in the environment. First, the ability to boundless expansion, and second, the ability to purposeful behavior. The same Japanese team demonstrated this difference between living cell and the probiotic models. Probiotic models in this picture, various kinds of the models, living cell, and between them, mysterious evolution jump or the missing link. Let's discuss the possible reason of such difference between these types of natural systems. In this picture is shown probiotic evolution directed to complication from dissolved organics via self-assembly to the following protocells and the complex artificial cells. And this is biological evolution. Both types of evolution proceed with the tendency to complication. So this biological evolution looks like a natural continuation of the prebiotic evolution. However, if we consider thermodynamic microstate of these systems, that is determined by the balance between the entropy contribution and the energy contribution, the difference between them becomes obvious. Prebiotic chemical systems exist with tendency to rise of entropy in accordance with the second law of thermodynamics, while biological living systems, on the contrary, evolves with possessing excess of free energy and information in respect to entropy. So it follows that any kind of prebiotic models cannot be transformed into living units simply by means of boundless chemical complication. It is necessary to overcome the thermodynamic or negentropy barrier between these kinds of natural systems. According to the elaborated concept, such transition might occur only by means of regular inputs of free energy and information into the system that which are so frequent and strong that entropy has not sufficient time to fully compensate them. And in this case, such a prebiotic system occupies the intermediate position between models and living units, and it is shown in the next slide. Prebiotic model, living unit, and very intensive processes with rise of entropy and rise of energy. And there exists a very strong competition between these processes. Such a system is intermediate between non-living and living, and it can evolve in the both directions. So it follows that the transition to life might occur only in highly non-equilibrium medium with the multi-level oscillations of physicochemical parameters. Such conditions are usual in hydrothermal systems. As an example, this is a record of pressure dynamics in Mutnovsky hydrothermal system in Kamchatka. We can see irregular macro fluctuations of pressure, regular migrants, and higher frequency oscillations are also present. And following these thoughts, the most suitable area for the emergence of life seems the upper zone of hydrothermal. This is a hydrothermal channel, and entails pulsating migration of fluid to the surface. From time to time, there arises the concentration of organic matter, and there arises a cluster of probiotic microsystems which interact to each other due to oscillating environment. And I consider such a system as a precursor to primary populations of the scheme of how such a microsystem can transit into life is shown the next slide. The next scheme, probiotic microsystem, strong fluctuations outside that influences the microsystem, red arrows arising of counteraction to the external stress that it follows of the Le Chatelier principle. And under near equilibrium medium, such counteraction, red arrows, is weakened. However, in case highly non-equilibrium medium, that means microsystems first are in the state of bicyclic transition from the initial stable state to new stable state, and high frequency changes in the medium that initiate, induce extremely fast chemical reactions. Such a system sometimes can become intensified response of the system to external stress can become intensified in comparison with the force of external influence, which means overcoming the negentropy barrier. And such a system has over entropic excess of energy and information, as a result of which it becomes active with respect to the environment. The system is already capable of expansion in the environment through the enhanced and targeted response to periodic stress. Such approach can be checked in the course of laboratory experiments of new times. Various types of probiotic microsystems, it's necessary to subject various types of probiotic microsystems by pumping with different fluctuations of physicochemical parameters. And the stress response should be investigated, especially the signs and changes that leading to life. For instance, it's possible to determine the rising of active transport increase of energy gradients and gradients of concentration. It's possible to check rise of homogeneity rate and so on. And the conclusion, two required components are necessary for the origin of life. First, prebiotic organic microsystems of optimal composition, that is lifeless matrix, and second, periodic high-frequency stress for pumping. And as a result of this process, this is final conclusion, a primary living microorganism or population integrates through an effective response to periodic stress. They appear surplus or entropy free energy provides its ability to expansion in the environment.
Okay.
While the
Vladimir,
surplus, yes.